Hello everyone. I'm going to tell you today about some work I've been doing in collaboration with Anil Agarwal. The work is spearheaded by a shared graduate student of ours, Tanya Barrera, with some help in the lab from two other of my students, Marie-Laure Sotolo and Kevin Kwok. And the work concerns the evolution of behavioral, sometimes referred to as sexual isolation. Now, it's often recognized that behavioral isolation is a first step in initiating the speciation process. We'd really like to understand how it evolves. And a long history of lab evolution experiments have shown that it at least sometimes evolves as a byproduct of divergent ecological selection. That is due to adaptation to different abiotic environments in a process referred to as ecological speciation. But the details of this process are not fully understood. For instance, are some axes of environmental differences more conducive to the evolution of behavioral isolation than others? Are there certain environments that make the evolution of behavioral isolation more via other processes more or less likely? We realized that we were sitting on a long-term evolution experiment that might be able to shed light on these. The design of the experiment is very straightforward. From a single Drosophila melanogaster lab population, we derived 28 replicate populations. These populations were placed in one of two novel larval environments, LE1 or LE2. These environments varied in food and temperature. And then within each larval environment, populations experienced one of two adult mating environments, either a simple standard Drosophila vial or what we call the more complex environment, which is a larger cage, lower density of flies with added structural complexity like multiple food sources and pipe cleaners for places to hide. We know from previous experiments that populations have adapted to these different novel larval environments. We also know that these mating environments alter sexual interactions. Mating rates are much higher in the simple environment than in the complex environment, for instance. And we know that populations have evolved in response to these mating environments. For instance, males from the complex environment have evolved to be less harmful to females than males from the simple mating environment. So we can gain insight into how these larval and adult environments affect the evolution of behavioral isolation by doing mating trials among different pairwise combinations of these populations to assay behavioral isolation. Now, with 28 populations, there are 378 different combinations, pairwise combinations, so it's clearly logistically infeasible to test all of these. So we took a targeted approach, using 3 different assays to gain insight into the role of the larval and adult mating environments in the evolution of behavioral isolation. And we were particularly interested in these mating environments because we know they affect mating interactions, and so they may play a key role in the evolution of behavioral isolation. So how might mating environments generate behavioral isolation? Well, there's 2 different possibilities here. One is that they may simply be an axis of divergent selection, which can drive ecological speciation directly. Adaptation to different mating environments generates behavioral isolation as a byproduct. Alternately, mating environments may indirectly affect the evolution of behavioral isolation if some mating environments are more conducive to the evolution of behavioral isolation, or other ones are more constraining. They prevent the evolution of behavioral isolation. So as an example, imagine a mating environment which has a high risk of predation. This may place constraints on the diversification of extravagant sexual display traits that hampers the evolution of behavioral isolation. Whereas in a mating environment with a low risk of predation, those constraints are absent and behavioral isolation can more readily evolve via whatever process is driving the divergence of those sexual displays. So to differentiate between these 2 scenarios, we conducted our first experiment. This experiment was designed to test whether behavioral isolation was stronger between pairs of populations that evolved in different mating environments compared to pairs that evolved in the same mating environment, as you'd expect under scenario 1. So we assayed 196 different combinations of populations using a multiple choice mating trial in which 30 males and 30 females from each of 2 populations were placed together in a cage, and we recorded who mated with who. And in every one of these mating trials, the 2 populations came from different larval environments, one from larval environment 1 and one from larval environment 2. Now in half the trials, the 2 populations came from the same mating environment, either both came from simple or both came from complex, as shown by the red comparisons. And in the other half of the combinations, the 2 populations came from alternate mating environments. Alternate mating environments, as shown by the blue comparisons. So if adaptation to the 2 different mating environments was driving the evolution of behavioral isolation, then we should see it in these blue comparisons, but not in these red ones. Now, results were not consistent with that first scenario, that is, mating environments directly driving the evolution of behavioral isolation, because the strongest behavioral isolation we saw was actually between pairs of populations from the complex where they're both in the complex mating environment. So here we have an index of assorted mating where 0 is random mating and positive values indicate positive assorted mating, that is behavioral isolation. And as I said, what we see as the strongest behavioral isolation on average was between pairs of populations, both from the complex. So no support for scenario 1, in which it's adaptation to different mating environments that's driving the evolution of behavioral isolation. And in fact, there's support for scenario 2 here in that all combinations that involve at least one population from the complex environment showed some behavioral isolation. And in fact, when they both came from complex, it was strongest, suggesting that there's something about the complex mating environment that's more conducive to the evolution of behavioral isolation, whereas something about the simple environment that is not conducive, that's constraining on the evolution of behavioral isolation such that you don't observe it. So if the complex environment is more conducive to the evolution of behavioral isolation, we'd still like to know how it's evolving in that environment. There's two possibilities here. In a conducive mating environment, behavioral isolation could evolve by a process of intersexual coevolution, whereby sexual selection or sexual conflict drive populations or cause populations to follow unique evolutionary trajectories. So in that case, behavioral isolation would be expected between any and all pairs of populations, independent of their larval environment, as long as they're from a conducive mating environment. Alternately, behavioral isolation may be evolving as a byproduct of divergent selection arising from some ecological difference other than the mating environments, like in our case, the different larval environments. So the expectation in this case would be behavioral isolation only between populations adapted to different larval environments, not between populations adapted to the same larval environment, again, as long as they're all from a conducive mating environment. Now, we can't differentiate between these two possibilities from our assay 1, because in that assay, population pairs always came from different larval environments. So our second assay was designed to test between these two possibilities. So we conducted 224 additional multiple choice mating trials of the same design as in the first assay, but we focused only on the complex populations. So both populations in each pair came from the complex environment, and half the time they were from the same larval environment, either larval environment 1 or larval environment 2, and the other half of the time they were from different larval environments. So if behavioral isolation was evolving due to a process of intersexual coevolution, we'd expect it in all types of these comparisons. Whereas if behavioral isolation was evolving as a byproduct of adaptation to the larval environments, then we'd expect it only in the blue comparisons. Here again, we're focusing only on the environment, the complex environment that is conducive, based on assay 1, to the evolution of behavioral isolation. And what we found were results inconsistent with scenario 2A, intersexual coevolution. That is, there was no evidence of any assortative mating just between pairs of populations when they came from the same larval environment. But there was evidence of positive assortative mating, behavioral isolation, when the pairs of populations came from different larval environments. So support for scenario 2B, that is, adaptation to different larval environments, generates behavioral isolation as a byproduct. Now, there was this unusual color effect in this assay. I'm not going to go into details, only to say that to tell who's mating with who in these multiple choice mating trials, it takes two different colors of food coloring. And we did this in a balanced way. And although what combination we colored individuals didn't generate any behavioral isolation on its own, it seemed to make one particular combination the behavioral isolation that was otherwise present go away when you're looking at populations adapted to different larval environments. Now, importantly, there were no coloring effects in experiment 1. The second experiment used different food coloring. It was done by different students in a different lab. But nevertheless, it made us nervous enough that we wanted to repeat this assay using a different technique and not the same coloring. So we conducted assay 3. Assay 3 involved female choice mating trials, not multiple choice. So in each case, an individual female for one population was given a choice between one of her own males, a homospecific male, and a second male, a heterospecific male. And half the time, the heterospecific male came from a different population, but the same larval environment, as shown in the red comparisons. And half the time, the second male came from a population from the other larval environment. So mimicking the design of assay 2. And what did we find? What we found were results entirely consistent with assay 2. So when females were choosing between her own homospecific male and heterospecific male from the same larval environment, she showed no preference for one male over the other. The frequency of mating with her own type of male was 50%. Whereas when females were choosing between her own male and a male from a heterospecific male from a different larval environment, she showed a significant preference for her own male, mating frequencies greater than 50%. So no support for scenario 2A, intersexual coevolution, because no behavioral isolation here. Support for scenario 2B, ecological speciation between larval environments, because there's a preference for her own male in these comparisons. The previous color effect was impossible in this case because females were unmarked. So we've confirmed the previous results using an entirely different assay. So let's put all this together from the 3 assays. What we've shown is that there's no evidence that adaptation to different mating environments was directly driving the adaptation of behavioral isolation. That is scenario 1. Adaptation to different larval environments was driving the evolution of behavioral isolation, scenario 2B, but this was only occurring in the complex mating environment. That is, the complex mating environment was more conducive to the evolution of behavioral isolation, whereas the simple mating environment was constraining the evolution of behavioral isolation. So the million-dollar question is, what differs between these 2 mating environments that's making one constraining on the evolution of behavioral isolation and the other permissive to it? We don't know, but we can speculate. We know a lot about the way interactions unfold, mating interactions unfold in these environments. And for instance, what we do know is that in the complex mating environment, mating rates are much lower. Females seem to have a lot more control over mating interactions. They can simply escape or flee if they don't want attention from a particular male. They can seek out an alternate food source where they're not being courted or harassed. And so female preferences may be expressed, they may matter, and divergence in them may generate behavioral isolation. Whereas in the simple mating environment, mating rates are much higher, and female preferences may be largely overridden. Females are constantly harassed by males, they have nowhere to escape, and so they end up mating at a much higher rate than is adaptive. And most of the mating interactions, in terms of determining parentage, may be post-copulatory, things like sperm competition. And so female preferences may not be expressed, and they may not be able to generate behavioral isolation. Now, testing these would require further experiments, but that's an intriguing possibility. So to end, I want to thank all the many Drosophila helpers in the lab and some funding sources, and Antoine Morin for photos. Thank you.